Dear friend, in this particular module, we are going to see the sample size for the clinical studies. What are the sample size? What is the significance of sample size and how it is being used for the clinical trials? What is sample size? Why sample size is required for a clinical trial? The sample or the study subjects or patients who adequately represent the population from where they are drawn so that true inferences about the results are obtained. Sample size in any clinical trial is a critical issue because it determines whether the trial has adequate probability or power to get significant results. And at the same time, larger sample size involves increased cost and increased concern about ethical issues. The sample size is one of the basic steps in planning any clinical trial, and any negligence in its calculation may lead to rejection of the true findings, and false results may get approval. With smaller sample size in a study, it may not be able to detect the precise difference between study groups, making the study unethical. Moreover, the results of the study cannot be generalized to the population as this sample will be inadequate to represent the target population. On the other hand, by taking larger sample size in the study, we put more population to the risk of intervention and also making the study unethical. It also results in wastage of precious resources and the researcher's time. Now we are going to see the components of sample size or sample size calculations. First, we are going to see about p-value or alpha. What is p-value? P-value is also known as level of significance, and in every clinical trial, we set an acceptable limit for the p-value. We assume p-value is less than 0.05 is significant. It means that we are accepting that probability of difference in studying target due to chance is 5%, or there are 5% chances of the detection in difference when actually there was no difference exist. In the other way, we called as false positive results. This type of error in clinical research is also known as type 1 error or alpha. Type 1 error is inversely proportional to the sample size. The next component of sample size is power. Sometimes we may commit another type of error where we may fail to detect the difference when actually there is the difference. This is known as type 2 error that detects false negative results, exactly opposite to the mentioned above, where we find false positive results when actually there was no difference. To accept or reject null hypothesis by adequate power, acceptable limit for the false negative rate must be decided before conducting the study. In another term, type 2 error is the probability of failing to find the difference between the two study groups when actually a difference exists, and it's termed as beta. By convention, maximum acceptable value for beta in biostatistical literature is 0.2 or 20% chance that null hypothesis is falsely accepted. The power of the study then is equal to 1 minus beta, and for a beta of 0.2 the power is 0.8, that is 80%, which is the minimum power required to accept the null hypothesis. Usually most of the clinical trial uses the power of 80%, which means that we are accepting that 1 in 5 times we will miss a real chance, that is 20%, that we will miss a real chance, real difference. The power of study increases as the chances of committing a type 2 error decreases. Next component of sample size is the effect size. The effect size is the minimal difference that investigator wants to detect between study groups and is also termed as the minimal clinical relevant difference. Effect size can be estimated by three techniques, that is through pilot studies, or number two is previously reported data, or educated guess based on clinical experience. Example for effect size. For example, a drug X is in a reduction of mean serum glucose level by 25 mg per deciliter, and drug Y results in a reduction of mean serum glucose level by 50 mg per deciliter. Here the absolute effect size will be 25 mg per deciliter. So the relative difference or reduction by a drug treatment is 25 by 50 or 50%. The for larger effect size, smaller sample size would be needed to prove the effect, but for the smaller effect size, sample size should be large. The next component of sample size is the variability. Variability is the population variance of the given outcome variable, which is estimated by means of standard deviation. Standard deviation can be an estimate obtained from the information in the previous studies because the variation is usually an unknown quantity. For a homogeneous population, we need a smaller sample size as variance or standard deviation will be less in the population. What is homogeneous population? When you consider a body weight range of 40 kg to 130 kg in a study group, then it is non-homogeneous. That means there will be more standard deviation. You have very low weight as well as very high weight. The range is very huge. And when it comes to a homogeneous population, that is narrowed down, the weight range is narrowed down to 60 to 80 kg, then you need a smaller sample size. This is how the standard deviation will influence the sample size. The final component of the sample size is the dropout rate. Dropout rate means that estimate of a number of subjects those can leave out the study or the clinical trial due to some reasons. Normally, the sample size calculation will be given a number of study subjects required for achieving the target statistical significance for a given hypothesis. However, in clinical practice, we may need to enroll more subjects to compensate for this potential dropout. If n is the sample size required for as per formula and if d is the dropout rate, then adjusted sample size n1 is the n divided by 1 minus d. Prevalence rate or underlying event rate of the condition under the study in population is very important whilst calculating the sample size. Sometimes we have to readjust the sample size after starting the trial because of unexpectedly low event rate in the population. Here is the classical example of sample size calculation. A study is planned to check the effectiveness of the dexmedetomidine in preventing post-operative shivering. From previous studies, it was found that incidence of post-operative shivering is 60%. Reduction in incidence of shivering up to 20% will be considered significant for the effectiveness of the drug. Then what would be the sample size for the study with alpha value that is 5% and power of 95%. So here the parameters for the sample size calculation are given. P1 is 60%, P2 is 20%, effect size is 40%, and Z alpha is 1.96. Z1 minus beta is equal to 1.64. Finally, based using this formula, the sample size calculated to be 40% to be included in each group of the study. So far, we have seen the general aspects and components of the sample size calculation and typically how a sample size is being calculated for a clinical study, that is a clinical trial. Now we are going to see specifically on the sample size calculation of the bioequivalence studies. Bioequivalence trials aim to demonstrate that 90% confidence interval of the test to reference ratio of the PK parameters between two formulations is fully included in the acceptable interval. Traditionally, the sample size of BE trials is based on power calculation and inter-subject variability and the T/R ratio of the product. Since the exact value of T/R ratio is not known prior to the trial, it is often assumed to be the difference between the treatment does not exceed 5%. Hence, uncertainty about the T/R ratio is expressed by using a fixed value for the sample size calculation. What is the importance of sample size in a BE study? Though we have seen the importance already in the earlier slide, to recap that for specifically for the BE study, the size of the sample is very important for getting accurate statistically significant results and running your study successfully. If your sample size is too small, you may include a disproportionate number of individuals which are outliers and anomalies. Thus skew the results and you don't get fair picture of the whole population. If the sample size is too high, the whole study becomes complex, expensive and time consuming to run. And although the results are more accurate, the benefit don't outweigh the cost. Specific to bioequivalence study, what are the factors influencing the sample size estimation? So we already seen that the factors includes the potential risk or sponsor risk of probability, that is type 1 and type 2 errors, power of the study, then the coefficient of variation, then the test to reference ratio. What is the patient risk or participant risk or type 1 error? In BE studies, the null hypothesis is that the test is not equivalent to the reference. If the null hypothesis is incorrectly rejected, which means if the drug was indeed not bioequivalent, shows that it is bioequivalent, then it is a patient who is at risk, and this is known as type 1 error. That means the drug product is not bioequivalent actually, but in the study, it shows that bioequivalent. So that error is called type 1 error. So this error is called as alpha, and this is conventionally set as 0.05 or 5%. Next is the sponsor risk or the type 2 error. If the null hypothesis is incorrectly accepted, and that is the drug which was actually equivalent but failed by equivalence, then it is sponsor risk. They cannot market a good drug. This is type 2 error or beta error. Beta is used to define the power. Beta is usually set at 20%, which means the sponsor can take no more than 20% chance to have a null hypothesis falsely accepted, and that there is 1 in 5 chance that the study might fail just by chance. The power of the study is usually set at 80%, which means 1 minus beta is equal to 80%. The study should have 80 or 90% power to conclude bioequivalence between the two formulations. Now, what is coefficient of variation, with how it influences the sample size for the calculation during a bioequivalence study? The coefficient of variation shows the extent of variability of the data in sample in relation to the mean of the population. Sample size also depends on the magnitude of variability and the design of the study. Various estimates to determine the number of subjects for a specific drug can be obtained from relevant published literature or published studies. The final and last component to calculate the sample size for the BE study is t-by-r ratio. The exact value of the t-by-r ratio is not known to prior to trial. It is often assumed to be the difference between the treatment does not exceed 5%. Hence, uncertainty about the t-by-r ratio is expressed by using a fixed value for the sample size calculation. We propose to characterize the uncertainty about the t-by-r ratio by a normal distribution over the log t-by-r ratio with an assumed mean of log theta is equal to 0.00, that is theta is equal to 1, and a standard deviation of mu which quantifies the uncertainty. Evaluating this distribution leads to statistical assurance of the BE trial. How you present the sample size justification in the protocol? The method by which the sample size is calculated should be given in the protocol together with the estimates of any quantities used in the calculations, such as coefficient of variation, power of the study, expected t-by-r ratio, and type 1 error. The basis of this estimation should also be linear. This is important, therefore, not only the sample size, and the sample size justification is an essential part of the protocol. This particular slide specifically showing the formulas used for the sample size calculation of the bioequivalence study. This particular slide shows the sample size calculation for the average bioequivalence study using the tabular format given in the published literature. Here you can see the variations, t-by-r ratio, and the power based on which the sample size are being calculated for a 2-period and 4-period study. In this slide, we are seeing the different literatures or different tools used for the sample size calculations for the bioequivalence study. So these are the sample size table which is provided in the Diletti et al. Published literature, the first one, and the second one is the software which is used to calculate the sample size, like SAS and R software. Then there is a free tool which is also used to calculate the sample size calculation for a bioequivalence study. In this slide, we are seeing the recommended minimum sample size by the various regulatory agencies for bioequivalence studies. In case of WHO, European Union, Canada, the minimum sample size is 12. And in case of US FDA, a pilot study that documents the bioequivalence can be appropriate provided its design and execution are suitable and a sufficient number of subjects have completed the study. The minimum sample size is 12. In Russia, it is 18. And for South Africa, for modified release formation, the recommended sample size is 20. For Saudi Arabia, it is 24. For the Brazil, it is 24 for a pivotal study. And Japan says sufficient number. Don't specify any specific number. What is the maximum sample size? In New Zealand, if the calculated number of subjects appears to be higher than it is ethically justifiable, it may be necessary to accept a statistical power which is less than desirable. Normally, it is not practical to use more than about 40 subjects in a bioequivalence study. So it is clearly defined in New Zealand that it is not practical to use more than 40 subjects for a bioequivalence study. In other countries, other regulations, it is not specified what should be the maximum sample size. In ICH E9, section 3.5 applies. The number of subjects in a clinical trial should always be large enough to provide the reliable answer to the questions addressed. With this, we are concluding this particular module about the sample size, its calculations and its significance, its components with respect to the clinical trial and the bioequivalence study. Thank you.